Can you hear me? So, hello and hi everyone. It's a privilege to be here tonight at ISFIT, and I can only thank May-Britt Moser, our award recipient tonight, for choosing to give her lecture at ISFIT in Trondheim in Norway. ISFIT was not on my radar or on the radar of any of my colleagues and friends that I've spoken to, and that's quite amazing considering it's 35 years old and the world's largest student festival. But I can tell you, since we've been here, the more we've seen and experienced of ISFIT, this is the EMBL team who's here, the more impressed we are. We have been working for the last month, weeks, days with the most incredibly accomplished and calm and collected person, the ISFIT president, Aurora Kolstad. Could we give a big clap for Aurora? Thank you, Aurora, for hosting us here and for ISFIT. And ISFIT, I have to really talk about this phenomenal organization because it is run by 450 student volunteers. It is professional and they are serving on so many different levels. It really is a model for the future. And not just that, but really ISFIT works within this student society here in Trondheim. If you have not been to Trondheim in Norway, really, or if you have students, children who are students, you must send them here. This place is incredible. A labyrinth of bars and saloons in this building alone. On Sunday, we were at the Peace Prize, and this really restored our hope for humanity. So really thank you, ISFIT in Norway. And honestly, if there's anything I can do to spread the word about this incredible place and to increase the number of students coming here, let me know, count me in. So who am I? I'm Menush Rayner, you can call me Nush. I lead alumni relations at EMBL. This is an intergovernmental institute with 30 member states, one of which is Norway. And you will hear more about EMBL from my colleague Cornelius Gross, who will be speaking later tonight. What I can tell you about it is that I work every day with thousands of brilliant life scientists who are exploring the mysteries and the secrets of life on a molecular level. And that's at EMBL and across the world because I also work with alumni. So talking of brilliant life scientists, I'd like to tell you why this is called the Kafatos Lecture. This is named after Fotis Kafatos. Fotis was a Greek scientist who was hired from Harvard to lead EMBL as its third director general. Fotis was passionate about communicating science. He set up the EMBL Communications Department, and he also set up Alumni Association at EMBL, and our alumni communicate science more broadly. He was also passionate about supporting young scientists, and so it's actually in tribute to him, he died in 2017, that this lecture brings groundbreaking life sciences to young students and to the public. So that's Fotis. This is our fourth lecture. The first was in Crete, the second was in Munich, and then Amsterdam, and now here in Trondheim. We go to where the scientists who are selected as award recipients, we go to where they are. We love this lecture series. It has given us the opportunity to be exposed to world-class, incredible scientists like May-Britt Moser, who do fantastic research, and we're very excited about bringing this further to the world. Tonight, this is being broadcast, this is streamed. We have about 1,000, well, we had 1,000 registered people online. We've got about 200 people here in the auditorium. And this will be available on YouTube. So feel free and feel welcome to watch it and rewatch it and to share it after this. If you'd like to nominate a scientist for the Kafatos Lecture, the eligibility criteria is that they should have received an ERC grantee at some point, an ERC grant. ERC is the European Research Council, which was founded by Fotis Kafatos, and he was its first president. So that's about the lectures. Now to some well-deserved thanks. Again, thank you to May-Britt Moser for accepting to give this lecture. Thank you to ISFIT and Aurora for hosting us. Thank you to a beautiful head of communications at Kavli, Rita Nilsson, for being our inroads into reaching May-Britt, and to the fabulous teams at EMBL who have partnered with us from day one. This is the EMBL Photo Lab, whose head is here, Doris Panay. The EMBL Science Education and Public Engagement, whose head is here, Agnes. She will do the Q&A later. We've got the EMBL Communications. They're not here, but they have been responsible for all the wide publicity for this, and whose design team have created the visual design here. That's Daniela Valesco in the team of Tabia Rauscher. And of course, there's the international relations team. I've got my colleague here, Sarah. Together with international relations, we try to bring science to our European member states. So through that, we have our council delegates and partner institutes here too, directors of. And finally, our own team. We're a team of 3 in alumni relations, but I would like to point out, highlight one, who is Tom Furnival-Adams. He's the person who takes care of all our awards, and this is one of them. So thank you, Tom. And of course, we don't do it alone. We have 10,000 alumni worldwide, and it's through our alumni in different working groups and committees that this lecture itself was even conceived by one working group. Then we have a nominations group to find great scientists. We have a selection group. We have a group that helps with fundraising. And on that note, we'd like to thank the Bodhisaki Foundation, who are a Greek foundation in Athens. They have funded this. We're really proud to be partnered with them because part of their mission too is education and health. So now all that's left to say is that enjoy the lecture. And since the theme of ISFIT this year is power, I can just say, may the force be with you. Thank you.
And thank you so much for the kind words, Noosh. I'm a student at the Norwegian University of Science and Technology, but most of my time I actually spend in this glorious house, organizing the International Student Festival in Trondheim, also known as ISFIT. We're on our 5th day of 10 days of ISFIT, where over 400 students from all over the world are visiting us. ISFIT started out back in 1990, after the fall of the Berlin Wall, to unite students across borders. And that's also what we do today. 111 countries are represented here during ISFIT. And this year's festival is about the theme of power, how it influences societies, and how we as students can drive positive change. Through workshops and also an extensive cultural program, which we are honored to have the Kafatos Lecture join, participants examine power structures and how they impact the world. And one of the greatest powers in our society is knowledge, the ability to seek truth, to question, and to discover. Academia and research fuel progress, yet it remains inaccessible to a lot of people. That's why hosting the 2025 Kafatos Lecture together with EMBL is a great, great honor. We are, of course, immensely grateful to EMBL, but we are also immensely grateful to May-Britt, who has chosen us as the venue for this lecture. May-Britt knows this house because she is an honorary member at the Student Society, and we're very happy to have her here today. On a more personal note, I still remember dancing on the top of the tables in my classroom in 7th grade when May-Britt and Edvard won the Nobel Prize. That's a long time ago, but it sparked an immense curiosity in life sciences for me. And I knew it sparked curiosity in a lot of other people as well. And for this lecture today, I hope it ignites curiosity in this international and young audience. Yeah, I'm very, very grateful for this. So thank you to MBL and thank you to May-Britt. Have a great lecture.
Okay, well, it's wonderful to be here. Thank you very much, Aurora. Thank you very much, Noosh, for welcoming us and starting off things. My name is Cornelius Gross. I'm a neurobiologist. I work at EMBL. I'm the director of its site in Italy. And in a few moments, I want to introduce and ask our award winner to come up and give the lecture. But I thought I would take a few moments to tell you a bit about the institution that's behind this award. Some of you know it, but many of you may not know it. So let me see if I can get slides here. Super, thank you. So EMBL is an intergovernmental organization. I think you heard that. We both carry out research, but we also foster research and provide services to the wider research community across Europe. And we were founded 50 years ago. We're a distributed institution, so we're actually located at 6 sites across Europe. We have 2 large sites. They're located in Germany, in Heidelberg, where the work focuses on cell biology and genomics. And we also have a large imaging center that uses advanced imaging tools and provides those to the community for the community service. We also have our second largest site, which is in Cambridge in the UK. It's also called the European Bioinformatics Institute. There they carry out research in bioinformatics and in computational biology, but also primarily what they do is they actually manage and curate and then offer to the community molecular databases. And probably if you are life scientists, you've probably used these databases before. They're critical part of what life science researchers do. We also have 3 smaller sites in Italy, where I'm from. We study epigenetics and neurobiology. We have 2 sites in Hamburg and Grenoble, which are focused on structural biology. They're associated with the synchrotron sources there, and they also carry out extensive sort of technology development, develop tools, set up the beamlines, actually develop robots around them that people then use who come and visit those sites to carry out their research. And then finally, we have our youngest site in Barcelona. It's on the beach. It's really beautiful. And there they focus on tissue biology, working with organoids, trying to advance in vitro cellular systems to understand organs and tissues. And also they have an important initiative in the area of theoretical biology, so trying to bring mathematics and mathematicians into biology to help us understand and actually bring a mathematical understanding to biology. So at its core, EMBL is focused on actually carrying out basic kind of blue sky, bottom-up research. About 50% of our staff are involved in that, and then the other 50% of our staff are involved in scientific services. So we actually develop services, we develop technologies, and provide those to the community. And this idea of mixing research and service together kind of in the same place, literally in the same building, in the same hallways, is a feature that EMBL has had from the very beginning, and the idea is that research benefits from having those service and technology people around and vice versa as well, to try to keep both kind of at the cutting edge. And just to kind of bring home how critical that is, actually the accesses to the databanks that EMBL manages at its British site have about 170 million hits each day, where people are asking for something from EMBL or from EMBL's databases. EMBL also has a major role in training young researchers, and we have a couple of features, the way we're structured, that promote that. So on the one hand, we have a limited term contract system, so people come to EMBL for a limited period of time, maximum 9 years. And then also we provide internal core funding. And this gives a kind of special atmosphere in which there's no competition between scientists there. Everybody has the same deal. People know they're only there for a short period of time. And it's also the reason why many people who have left EMBL, all the 10,000 people you heard about, the 10,000 alumni, are actually typically then returning to the national systems to contribute to research inside of our member states. And finally, EMBL is involved in translating its research findings, its technology development, obviously, that's very important. And then it does have a special role in advocating for funding and also for policy changes at the international level. So we have a special position to advise the European Commission on directions in life science, other international bodies, and as, for example, we heard just before, the European Research Council was in many ways a product of EMBL, having been founded by and then also led by Fotis Kafatos. So Norway is involved in 2 different institutional collaborations. Actually, it's quite unusual for a country to have 2 of these. One of them is we have actually a representative here today from these institutions. One is the Michael Saar Center for Marine Biology in Bergen, not far away from here. And then there's the Norwegian Center for Molecular Biomedicine, Biosciences and Biomedicine in Oslo. And actually, the latter institution is also an EMBL model institute in the sense that they have taken on some of the features of the limited term contracts and internal funding, which is particular to EMBL. And it's actually part of a larger Nordic partnership in which we have partner institutes in each of the Nordic countries. And this has actually been highly successful. One of the young group leaders in the Nordic Partnership institutions, Emmanuelle Charpentier, you may know her name. She actually won the Nobel Prize in 2020 for the discovery of CRISPR editing. And also the Finnish Institute for Molecular Medicine, for example, has risen rapidly to be sort of a key global leader in the area of population genetics, which is a really expanding area of life science. So there's a lot of excellence in the Nordic countries, and we've been privileged to be part of that and to partner with them. Norway and EMBL are also involved in various infrastructure projects, part of the ESRI system. If you've heard about that, there is the Elixir consortium, which is about trying to standardize data access and life science databases. There is the Eurobioimaging, which tries to bring advanced imaging technologies to the community. And this is something that's done together between Norway and us. And then finally, I wanted to point out some other collaborations, grants, and research. There, for example, has been over the last 2 years, a large expedition that was headed by EMBL, but actually really carried out in collaboration with 90 different institutes across Europe, and in particular with a number of research institutes in Norway to do sampling of soil and water all along the coastlines. And this obviously was very important. Norway has a very long coastline. It was actually done with a sailboat. So perhaps you heard about this, and these samples are now being analyzed. It was finished last year, where they finally finished the whole tour through the Mediterranean finally in the second year. And so Norway has been an important partner in that, and also were critical in the award of this BioOceans 5D award from the European Commission, which supported much of that project. Finally, just to mention, in the area of data sciences also, there's the Federated European Phenome Project. This is actually very important. It is working to try to, again, standardize how we actually set up hardware and software to make clinical data, human data, available to the research community. So you may have realized there's a very large push in many countries, particularly in the Nordic countries, to actually characterize, phenotype, sequence humans in the population. And of course, there are difficult questions there on how to make that data available so that researchers can also access it and actually learn something about biology from our data. And Norway has been also a leader in that area. Finally, just you may be asking, how can we be involved in EMBL? I just want to put up a few highlights here. First off, we run a well-known PhD program, international PhD program. So if that's of interest to anybody, please check out the website. We run applications twice a year. Also, we run 2 postdoctoral fellowship programs. One called the iPod Fellowship Program is intended to foster interdisciplinary research. You work between 2 different labs. One of those labs can actually be in one of the member states, and the other one is at EMBL. So it's a great way to connect what you might be doing or what you're interested in doing in the country where you are and working at EMBL. And the second one is something we started recently. It's been quite an interesting experience, actually. This one is focused on people who want to do 100% technology development. They don't necessarily want to do research. They want to develop technologies, whether that's hardware or software or algorithms or microscopes. And this is a special fellowship just for people like that who want to be in a research environment but want to build tools that will then someday be useful for the community. We host visitors. We have a sabbatical programs for various topics, so please check that out if that's interesting. We hold lots of courses and conferences, primarily at our headquarters site in Germany. We do a lot of this in collaboration actually with our sister organizations called MBO, not MBUL, but MBO. We do many things together with them. And then finally, also, we welcome people who are anxious to work in our administration or in our international relations or communications departments to take part in what we do, also on the level of interacting with international bodies and policy. So, I don't want to take more of your time. I would like to ask May-Britt if she would maybe come up to the stage. Thank you. May-Britt Moser, this year's winner of the Kafatos Award. Just a few words before I want to give you the actual award. May-Britt is a fellow neuroscientist. I'm sure you've heard about her work before, particularly because she comes here from Trondheim. And we heard already she's the founder, she's also the director of the Kavli Institute for Systems Neuroscience science here. In 2014, May-Britt was awarded the Nobel Prize in Physiology and Medicine for her groundbreaking research on how the brain uses grid cells, and we'll be hearing about that today, I think, with its musical interlude, to create this internal coordinate system. And she also had an essential role in showing how these are important for spatial navigation and for memory. And she continues to lead innovative research into brain mechanisms of learning and memory. And also throughout her career, she's been particularly instrumental in establishing and leading institutions that are collaborative and interdisciplinary in its nature, I think very much in line with EMBL's sort of philosophy. And she continues to be an inspiration to generations of neuroscientists and students, many of you of who are at this event today. So it's my special honor today to present you with this award on behalf of EMBL and the EMBL Alumni Association. Congratulations. Thank you so much.
Maybe we should go on. Thank you. Thank you.
So I think the floor is yours. It's your show. I'm looking forward to it. And thank you again so much. It's really our special honor to be here with you today. Thanks.
Thank you. And to see. Okay, so you fix it. So while I get help, which is excellent, thank you, I would just like to say thank you for the prize, the Kafatos Prize. I'm deeply honored, but and I'm also extremely thankful to ISFIT and Studentersamfundet for hosting this lecture. I just got this idea, I just need to give this lecture in the Studentersamfundet, and then simsalabim, you just organized it for me. And then I thought, because I was asked to give a popular lecture, and then I thought the best thing I can do then is to invite my band. And we have traveled around in the world with different stories. This story we've tried only once, I think for 1 hour or half an hour. So this will be extremely exciting to see if we work well together. I think we will. And I would also, of course, like to say thank you to the team who has helped me at Kavli. Thank you to NTNU and, of course, the lab. And just to tell you about how did I get this title. So I was so inspired by ISFIT, the power of knowledge. And one way to get a lot of knowledge is through science. And our question, our power of science, is that we think that together we can uncover the algorithms, the recipes that the brain is using to generate who we are, our thoughts, memories, ideas, and friendship, emotions, everything. This has been a question that has interested me since I was a child. And luckily, in the 80s, I met this man. In fact, I met him before because we went to the same class at high school. But here in the 80s, we decided to work together. And we were invited to the lab of Per Andersen in Oslo. And we were allowed to explore the brain together. And now, next year, it will be 30 years that we two have collaborated when we built our lab in the autumn of 1996. And we have had the most fantastic people working in our lab and still working with us. And as I say always, it's a great blessing for me to be allowed to work with these brilliant people. So when I have a bad day, I go to the lab and then, yeah, this is fantastic. So of course, the brain is big, and where should we start? And Edward and I decided that the hippocampus is a great place to start. So even before we were born, then there were results in the literature of humans who had epileptic seizures, and medicine didn't help them, so they had to remove these beautiful structures that you see there to the right. The left one, that is from the human brain. The right one, of course, from the seahorse. So it's called the hippocampus. And when they removed this structure from a human brain, this person was functioning better because the epileptic seizures stopped and was social, everything was fine, except they could not code new event memories. And they couldn't find their place. And the most famous of these patients, that was HM. So when you cut out the hippocampus, if that would happen to you ever, you will not remember that you've been here together with us, for example. In order to study this structure, we have to know how to do it, and we know that the nerve cells, the neurons in the brain, they talk to each other with electrical signals. There's some chemistry too, but the main important thing here for us now is this electrical signal. And why is that interesting? Because we can monitor the activity of such cells when animals, and yesterday I was at a lecture where they showed humans performing spatial tasks and memory tasks. Here we talk about animals. We have small sensors close to the cells. Just see if I find the mouse. Do you see the mouse? Yes. So you have these small sensors close to the hippocampus cells, and then while the animal is running happily around, and I have to tell you the slogan for our lab, we have excellent science, happy people, happy animals, and diversity. It's allowed in our lab to say diversity. Here is a rat. You listen to one such hippocampus cell, hopefully. Yes? Do you hear this? It's not a nice signal, but it's every single red dot there. That is such an electrical signal from one hippocampus cell. What you see here is that whenever the rat, Emma, is up in the northwest corner, then there is activity. But when she is in other places of this box, it's silent. And we can show this in another way with colors, and then hot colors is showing this very, very activity. Oh, I lost my pencil, but then I have this one. So we're going to demonstrate place cells for you, and the reason why is that there's, of course, not only one hippocampus cell that we should listen to. We should listen to many. And why is that? Try to find out what these cells are doing. Okay. Then we record from my hippocampus. And oh, I was at the wrong field. And then, so this place cell had activity here, you heard that. And another cell. And another cell. Thank you. So I think you understand that these cells, they have their private preference for different places in the room. And that is why one of my mentors suggested, John O'Keefe, who we got the Nobel Prize with in 2014, suggested that hippocampal place cells signal where the animal is. Edward and I went to London and we learned this from John, how to record such cells. And when we went home in 1996 to enter NU, then we had one question with us. And we really wanted to understand, where is this spatial information coming from? And then we decided, after many, many control experiments, beautiful experiments, that we had to record in the medial and entorhinal cortex. And this was done with a lot of people, so I should stop myself from telling you all these people, because then I will end up with only names that you don't know. But you see, I've cited the papers underneath here. So what we decided in this collaboration was to record them from entorhinal cortex. This is the back of the brain. The colorful map here, that is the entorhinal cortex. To the right, that is the medial entorhinal cortex, and to the left, you have the lateral. And in the medial entorhinal cortex at the top, which we call the dorsal medial entorhinal cortex, that is the place that's sending information to the cells that we just heard in the hippocampus. So when we recorded in this area, and people had been there before, and they concluded that, oh, there's nothing interesting here. But we had big area in our lab, so we had bigger boxes, so we could see something that people hadn't seen before. We saw this pattern. Here, what you see is the black traces. That is the trace of the animal, and there's a camera above so that we know where the animal has been. This is a recording from one cell. You remember the place cell had only one field. This cell has many fields. And what is so interesting here is that these fields, they are so regular. You see the colored patterns there, that is the electric potential that we saw with the place cells. This is also with the grid cells. That we can fill in equilateral triangles between each field. This is biology. It's not mathematics, and still it's so beautiful. So we decided that these cells had to be called grid cells because they are active in any environment, so it's resembling a universal coordinate system. I also have to tell you that these cells, they come in different resolutions. So at the most dorsal part, then it's fine resolution with small fields and a small distance between the fields, and then it's becoming larger and larger. Why is that interesting? Because, you know, when I said that Edward and I went to Trondheim to try to understand where the spatial information in the hippocampus is coming from, we recorded these cells. And if you just do a simple, this is a model, if you just do a simple linear summation, you just sum up the middle. At the edges here, it's just cancelled out. And what do you get? You get one field, like we saw. Maybe we can play one place cell. Perfect, thank you. So we found a potential code for where this information is coming from, how it's generated in the hippocampus. So there are 50 years of single cell coding for navigation in open space. You've learned now about the place cells. In the medial entorhinal cortex, you learned about the grid cells, and we decided that we wanted to illustrate this type of cell. That's called a head direction cell, and this cell is active in the compass directions, subjective compass directions. So if I record such cells from my entorhinal cortex, and we select a cell that is active in this direction, and then, thank you. And then maybe we have one more cell, just to illustrate. Thank you, Bagman. Beautiful. So each cell has its own preference for one direction. And then we have speed cells, and these cells, they signal the speed of the animal when it's running around. And then we have a very interesting cell, and that is telling us where the boundaries in the environment is. Thank you. And finally, we have cells that respond to objects in a certain distance from the object and also a certain direction. So we have all these kinds. It's like a jungle of cells. But do we understand more about the recipes when we know about these cells? We know that in the human brain there are more than 80 billion neurons. So in order to understand these recipes or the algorithms, we need to know how do these brain cells work together. But what is preventing neuroscience to move forward? That is tools, if they are available or not. It's not about the questions, but it's about tools. Do we have tools to let us record all these cells that we need? Yes, so we've been so lucky that we've been collaborating with people starting from Janelia and other places to join them to develop NeuroPickasus 2.0, which are silicon probes, small microchips that you can then use as sensors. Here we suddenly can record thousands of cells. To the left here, you see 149 grid cells of one size. And there were many, many, many other cells. Why is this interesting? Because suddenly we can start to ask not only how the activity looks like when we sample for 10 minutes, but we can ask of how does the activity look like at very, very small time bins. So we can talk about how the the population activity in sub-second time bins, like here. So here, on this figure here, here's cell 1, and then every single dot here is one pop, 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 pop. And then you have the second cell and third and so on, 149 cells. In this time bin, then it's the activity of the population. Why is this interesting? What is so nice is that activity in this area especially is correlated. And since it's correlated, we can use new mathematical techniques that we call low-dimensional techniques, so that we reduce the variability from 149 vectors or rooms down to 3 rooms. And then we have the ability to see patterns that we couldn't see before. So if you then imagine that this activity here is one dot in here, I hope you see that this is a torus. And the activity of the animal in the box is mapped on this torus. And why is this interesting? I can tell you very, very short about the head direction cell first. And you have learned about the head direction cells, but the model tells us that the connectivity between cells facing north should be connected to those to northeast and northwest, but not to south. Like you see the cat here, it can't move the ball out of the frame. So these manifolds that we call them are like frames constraining where the activity can go and where it can't. This UMAP is a way to visualize this, but we need to quantify it. And then we had to go to what we say here, to the GLUS, to the mathematical faculty, to learn how to do topology measurements. The topologists, they know, I don't know if they call themselves, our rector is mathematician, so you might nod when I say a topologist. But in any case, we call these mathematicians that. And they know how to describe a circle with such Betty numbers, and you can, it's a complicated method because you have holes and all these things. But if you just accept that a circle has these Betty numbers, these circles here, these dimensions, and a sphere has this formula, and the torus, the other formula, we can ask, with these methods, these mathematical methods, can we find a torus? And then you ask about this, I don't know if this works, yeah. A torus has two circles, and the long one you see here, and it's far beyond the noise in the background here. And then you have two circles, and then you have one circus in the different dimensions. And this is exactly what is needed to tell that this is a torus. This is when the animal is just running around in the open field. While the animal is running around on a wagon wheel, this maze that you see here, you don't see the grid cells that well, but still you have a torus. And you know what is so fantastic is that when we sleep, this is my grandchild, by the way, Alfred. He's sleeping. So when we sleep, either it's in the dreaming or the slow wave sleep, this torus is just running and running and running around. Isn't it amazing? So a toroidal code we could think of as an algorithm in the entorhinal cortex. And this was predicted by models before we got the data. These models are continuous attractor network models. And they suggest how the population activity is constrained within the manifold. And what we've learned is that our brains have this continuous internal map that is just running and rolling and rolling. And what is so fantastic is that this is independent of sensory information, since it happens when we sleep. So it's internally generated. Thank you. There's another secret that the grid cells hold on to. And that secret was published from our lab this year, in fact. And that is that if we see, just 1 second here, if you see just a rat walking around like this, and we decode the activity of the rat, we know exactly where the rat is if we don't care that much about the time resolution. But as I said, because we record so many cells, so many electrical potential at the time, we can do much finer analysis. We can go down and ask, how does it look like, for example, at theta cycles, which is around 8 seconds eight times in a second. And then we can ask, is it so that the position is still located around the rat, or do we see some look ahead activity? And we do. So what we see is that when the animal is going around in the environment, there suddenly sweeps outside to the right and to the left and to the right and to the left. And this happens 8 times in a second. And I can show you the video if it works. Yes. Thank you. You remember that grid cells had different types of resolution, and we can group the grid cells into different modules. What we see is that the different modules have different lengths of these sweeps, and that is shown in this figure here. So these sweeps might code the environment much more effectively than by just having a dot of the position of the animal. Because these sweeps, we've shown in different models that it's an extremely good way to cover the environment. It's like my brother said, oh, this is like sonars that we have on the boats at the West Coast. Until now, we've talked about recording these or monitor this electrical activity, but we haven't seen the cells. And we heard that in the EMBL, it's possible to go and build microscopes. We are building microscopes at our place with our fantastic researcher, group leader at our place, and I will show you the name afterwards. So the question here is, can we reduce a 500 kilo 2-photon microscope to be so tiny that a mouse of 25 grams can carry it and still be happy? You remember the slogan, excellent science, happy people, happy animals, diversity. This has been possible, and it happened first in Peking University. It wasn't ready. Wei-John came to our lab, and as a postdoc, and now, as I said, as a group leader, he developed this tiny, tiny 2-photon microscope so that the animal can run around, and you see this star-like activity, which is, you see directly into the brain, into the entorhinal cortex. And this brain, or this mouse, has been genetically modified so that the activity of these cells is possible to visualize. And this is a way to record many grid cells at the same time, thousands. So not only the electrical potentials, but here we see where the cells are and how it looks like. Thus, we have now at least 2 powerful tools to uncover the algorithms for higher cognitive functions of the brain. There's somebody who don't have. And you will hear now a sad story through music when you don't have what you need to function. Thank you. This is probably the first time you ever heard a cell dying in a music. Did you hear the sweeps searching through the empty rooms, finding nothing, finding no memories? And this is how terrible it is to have Alzheimer's disease. First, people start to complain about some memory problems. They don't find their way as easily as they did before. And if they go to the brain scanner and check their brains, if they see that the entorhinal cortex has been shrinked, then they are in real trouble. Because that is it's a good prediction, but it's a bad prognosis for developing Alzheimer's disease. It's dying cells in this region, and the first cells that die in this region, that is the grid cells that we just learned. And the symptoms, the diagnosis, is based on problems with remembering event memory and spatial navigation. So we, in science, we need, and not only in science, in society, because with a growing population, older population, we get more and more of this disease. So we need to understand why these cells die so that we can rescue them. And what you've seen today is that we can both record from these structures, and we can see the sick cells, and then we are closer, I think, to understand why they die and how we can rescue them. But we need more science throughout the whole world to solve this big, big problem. And I'm not the only one saying we need more knowledge. Jens Stoltenberg, our former Prime Minister, the former Secretary General of NATO, and now our Finance Minister, he said, knowledge trumps everything. It is each other's work and each other's knowledge that we will live on in the future. And then we will end. Thank you. Thank you so much. And thank you for being a great audience. Alle får deg på scenen. Jeg synes det blir...
Okay, we haven't finished. We haven't finished. This was just the end of the show of understanding the power of science and uncovering the algorithms of the brain. The next part is a Q&A with May-Britt, which Agnes will lead. There will be a very informal conversation, and people online as well as people here can ask questions. Before we start, May-Britt had asked me to thank the chamber ensemble, who was the Trondheim Solistene, as well as the composer Bertil Palmak Johansen, and of course the fabulous team at the Kavli Institute. So thank you to all of them. Okay, so over to you, Agnes.
Thank you so much for this amazing presentation, for a really unique interplay of sense and music, sense and arts. Both amazing tools being able to express the understandable and making knowledge amazingly accessible for us all. It's a pleasure to have you here tonight and help us understand how our conscious memories and our environment shape us and make us who we are. Because of course when we learn about the brain, then we actually learn about how we perceive the world, how we perceive our environment, and it's of course by being a highly scientific exploration. It's an amazingly deep personal exploration as well. I would like to invite you all here in this hall, and of course online as well, to take this wonderful opportunity to discuss some exciting questions with May-Britt concerning our brain and how it functions and what we can learn from her research. For those of you joining us online, we are delighted to have you with us, and please put your questions into the Q&A section on Zoom. We will make sure we include them in our conversation as well. And for those of you sitting here, please raise your hand if you have a question, and one of our colleagues will be walking to you with our mic.
Do you see anybody?
Yeah, that's exactly what I want to mention, that we're extremely blinded by the lights, so we won't be able to see any of you much better. Thank you very much. All right, lovely. Just to give you 2 minutes to think about your questions, if you don't mind, I would love to start the conversation with one of mine, and that is, May-Britt, where is your passion for studying, observing animal behavior come from? When did it all start? When was the first time you thought, oh, this could be
I think so. I was raised on a farm, and I loved animals. I think the day I was born, and since then I was so curious of why do the animals behave like they do? Why do humans behave like we do? And especially these days, we really want to understand why people behave like they do. And then to be allowed, as I said in my talk, to work with eminent, fantastic, creative people, to follow this passion that I had when I was a child. That is just such a blessing. So I'm extremely, extremely grateful for that.
And then maybe, and I already see questions coming in from the online audience. You mention in your different presentations and kind of publications a lot about the experiments, and today as well, that you've been running in the lab, and then of course yourself, and then with your team and the bigger teams. Can you maybe describe a little bit more about maybe one of your first experiments? Why is it so special to work in a lab and actually to design and run an experiment with animals? What makes that so special?
So, first of all, of course, we could have studied humans, but that is extremely challenging of many reasons. Also, even though all the studies, of course, are ethically beautiful doing human work, there are not so many patients that you can, for example, record from. And since we are so eager to understand the recipes, the algorithms in the brain, we need to record from, as I said, thousands of cells. And that hasn't been possible yet in humans. But I think what is extremely important is to have this respect for the animals that we use, and almost ask them for permission. And the reason is, of course, that they didn't ask to join the experiments, but they were allowed to. And in our lab, I'm so proud of the people that people treat them as pets. And that is why now every lab will share data, because you have to. But even before we had to share data, Edward and I loved to share data from our lab, because we knew that in our lab the animals have their best life, we have the best tools, we have happy people, and then when we give out our data, first of all, we know that it's excellent quality, and then we also know that others don't need to do those experiments. So because we have the 3 R's, and one of them is we want to reduce the number of animals that are in. But then when we have these few animals and we work with them, and I have a dog at home, and the rats are like dogs. They collaborate with us, and it's beautiful. So the pleasure also by working with these gorgeous animals is great, and then we get all this beautiful data. So I don't understand how it was possible that I was so lucky that I was allowed to do this together with these enormous, fantastic people.
Thank you. We are still a little bit blinded, but I wonder, okay. All right, great. Then we take the first question from here.
First of all, I wanted to thank you for the wonderful lecture, especially as a psychology student. It's been such fun to watch. And my question is, can we also visualize the next motion that the rat is going to take? So to kind of see the planning process, kind of predicting it, can we visualize that maybe in the near future?
Yeah, I don't see where you are. Can you raise it? Oh, okay. Thank you. Then it's easier to answer. So we sort of visualize where the animal is going with these sweeps, since they're pointing forwards. But it's a very interesting question. I know that I don't answer your question correctly, because you want to know if we can predict where the animal is going, but at least we can say or we can study what happens, for example, with the sweeps that is looking forward, if the animal is doing something different, like going backwards. Would the sweeps then point backwards or forward? And by doing all these experiments, then we can answer at least such questions. What is also so exciting, and I know you didn't ask about that, so forgive me for that, but I just have to tell about it. I find it so interesting, is that by looking at these cells, especially in the hippocampus, we can even tell which room the animal is in. And you heard about the Studentersamfunnet, there are so many different rooms. So if we had recorded cells from your hippocampus, we could tell whether you are in here or in different other rooms here at Studentersamfunnet, because the different cells are active in different environments, or the different maps. And we can even play with these things when it comes to odors. So regarding predicting where the animal is going, so if you train an animal to go to left when it's smelling chocolate, and go to the right when it's smelling banana, and if you then record the maps that are generated in the brain, you can in fact, just by looking at these maps, predict whether the animal is going to do an error, and if the animal is doing an error, guess how the map looks like. It's empty. And we know how the map looks like in these brain structures when the animal is going to left and to the right, just by being in one place and smelling the odor. So maybe, and also there are other experiments, I forgot about that, and that is, in the lab we have different kinds of environments and mazes, and we have a maze that looks like an 8, the number 8 shape, and when the animal is going in the middle, and if we record from the hippocampus, then we see that the cell will change its activity, the frequency of the activity, if the animal is going to left, and the same cell will then, so if it's decreasing the activity to left, it will increase the activity to the right. So again, we can then say the animal is about to go to left just by looking at the activity. So it's quite amazing. So you asked, why study this? Because we learn things that we didn't expect to learn. And it's just, what? Is it like this?
Thank you very much.
Thank you.
Thank you. Then let's take a question from our online audience. Have you made any studies with blind mice? Are grid cells also active in blind mice?
So we have not done any studies in blind mice, but so what we learned today is about the torus, and that is a kind of an internal map. But it needs to be anchored to the environment. And normal organisms, normal rats, normal humans, with eyesight, will use the eyesight in order to anchor the torus and then anchor the map to this environment. But we know that blind people are excellent in finding the place, so they have other ways to anchor the torus. So I would be extremely surprised if there are no grid cells in blind people and animals. So yes, and I get this question many times too. Is it so that those who can't find their way, if they don't have grid cells, if they're not sick, like having Alzheimer's disease, they have grid cells, I would say. It's just that they don't use it. Because you need to anchor your torus or your grid cells to the environment. If you don't pay attention to where's the stage in this room, where are the doors, your torus might just rotate and flicker around, and of course you can't know your way then if it's not stable.
And then maybe still talking about neurodegenerative diseases, we have a question asking, what's your opinion? Why is it the grid cells that are the first to die upon the onset of a neurodegenerative disease, such as Alzheimer's? Or can we say that because the grid cells die first, we see an enhanced effect of neurodegeneration?
So if I could answer that question, then we would be so close to be able to repair the cells. So I wish that I could suggest it. I know people working around in the world, even at our institute, because we also have a center for Alzheimer's disease and dementia, where people have some theories why especially those cells that we call stellate cells and fan cells in the entorhinal cortex, why they die first. But I don't know if I'm allowed to say because it's not published. But the nice thing is that people are working on these questions, and if we know why they die, then it's so much easier to rescue. Because there's so many techniques now. So say that there is a protein that is expressed too much or phosphorylated, then we can put in a virus and say, please don't phosphorylate this molecule, and then you can rescue the cells.
Okay.
And these viruses, of course, are kind viruses like vaccines, not dangerous.
So we are definitely very hopeful that we will have
and
soon.
Yeah, absolutely hopeful, and especially if we join forces throughout the world to go to this. So earlier, it has typically been one idea that has driven this research in dementia, and that is never good. You need diversity, also on ideas. And the way you get diversity on ideas is to have people coming from diverse places and education and everything to work together, and then suddenly they suggest to look for something that people didn't think about. And that is how the leap of faith is, that you make a jump in science, high risk, high gain, that you didn't expect to get results.
All right, that's very encouraging. Then we have another question here in the auditorium.
Hi, my name's Chetan. I'm an ISFIT participant from Canada. And you said something that I found really powerful in saying that it's not the questions that are limiting us, but the tools. In your opinion, what do you think are the most kind of novel and innovative tools that are being developed right now that can propel the field of neuroscience or human biology, biostats, biochemistry, what have you, forward the most?
Oh, thank you for that question, because then I have to correct myself. Because, of course, the questions are also extremely important. So our attitude in the lab has been that we never marry a technique or something because the questions we don't care about. Of course, the questions are the main driver of our science, but we can't move forward sometimes if we don't have the newest tools, because for example, like I told you about recording so many cells, that has been essential for us to dig into the algorithms of the brain. And I think that, again, I have to be careful because I know things that I'm not allowed to say, if you understand what I mean. I'm coming from a super lab and work with super group leaders at the Institute. So I can't tell all the secrets, but I can promise you that there are tools coming in the next few years that will just be mind-blowing, also from our place.
Thank you. Okay,
and we will share them, of course.
Mm-hmm. We have another question here. Yeah.
Hello. So I'm Cornelius from Liberia. I'm studying forensic science with concentration in forensic DNA analysis and interpretation. So I found your research fascinating. So I would like to know, how do genetic factors and developmental processes shape the brain architecture?
Oh, I'm a trained psychologist, and when we got exams, they always asked, how do we become the ones we are? And of course, it is, like you say, it's DNA, and it's the environment, and it's the interaction between our genes and the environment. And I think that if you think about the genes as all these beautiful recipes being there for us to use, to express the one we become. And then not all recipes are turned on. This is your field, this is not my field. But you understand what I say. And then some environments constrain what can be turned on, and some environments enrich. What I believe is that with knowledge and with science, we are able to find out how to not restrict our DNAs so that we can grow into people with no diseases or less diseases and being better human beings. We need so many good people around in the world these days when it's so many sad stories.
Thank you.
Thank you. Okay. Being, of course, conscious of time, unfortunately, we are getting to the end of our Q&A session tonight. Thank you so much for giving us such exciting insights into your research and the future of this field. I'm sure we all gained a lot of new perspectives about what's happening in our brains and how fantastic structures are there, and so much to discover in the future. And I hope we have a lot of young students here who will be very eager and keen to join the exploration in the future. We are delighted to share that next year's Kafatos Lecture will be given by Svante Pääbo.
Oh, wow.
And for more information about this lecture, and of course past lectures as well, please visit our website. So thank you so much again for this wonderful conversation and discussion. Thank you so much for your insight. We wish you a wonderful evening. Take care and goodbye.
Can I say something?
Yes?
Can we all raise up and say, hooray for science? We believe in knowledge. Yes.
Yes!